great.
Okay, great. So as Kenton mentioned before, I kind of get into vaccines specific for COVID-19. I want to give a little bit of kind of an immunology 101 to kind of get everybody on the same page as to what we're expecting a vaccine to do. Okay. So the immune system is roughly divided into two parts, innate immunity and adaptive immunity. And what we're really going to be talking about today lies in the adaptive immunity section, because the adaptive immune response is the part that has a memory of previous infections or memory of vaccination and can provide protection against future infections by the same pathogen. And so the adaptive immune system gets activated a little later in infection compared to innate immunity. And it takes about 7 to 10 days really for these adaptive immune components to start providing protection against a given infection. And so there's really two main cell types involved in adaptive immunity. There are the B lymphocytes and the T lymphocytes. The B lymphocytes are the ones that make antibodies. And so I'll talk a lot about antibodies today. And they are proteins that will bind to the virus specifically and neutralize it. In the case of a vaccine, hopefully neutralize the virus and block it before it has a chance to start an infection. And then T cells, we'll talk a little bit about T cells as well. There's two main types of T cells that we're interested in as far as vaccination. The first ones are helper T cells. We often call these CD4 positive T cells. And their role as helper cells is to help activate B cells, and so to stimulate antibody production from the B cells. The other types of T cells that we're interested in are cytotoxic T cells, and so we often call these CD8 positive T cells, and their job is to kill infected cells. And this is going to be particularly important in virus infections because viruses have to replicate inside of host cells. All viruses are carrying out their replication cycle inside of host cells. So the adaptive immune system, as I said, it has this specificity in recognizing specific pathogens, providing a memory response against pathogens we've been exposed to before or have been vaccinated against. And it's really trained not to recognize potential pathogens, but it's trained to not recognize self-antigens or antigens of the host. And so over here on the left is showing what's happening during development of adaptive immune cells, of lymphocytes. So as these cells are made in our bodies, they each have a single type of receptor that recognize a given antigen. So the term antigen is just the part of the pathogen that's recognized by a B cell or antibody or a T cell. And so as these lymphocytes are made, they can recognize any given antigen. And so the immune system exposes these developing lymphocytes to what we call self-antigens or proteins from our own cells and tissues. And then any of the lymphocytes that recognize those antigens are deleted so that we don't mount immune responses against our own antigens. And so what's left are these lymphocytes that have potential to recognize things that aren't part of self. And so then later in life, when you're exposed to a pathogen or a vaccine, noted as foreign antigen here, the given lymphocytes, B and T cells that recognize this foreign antigen then get expanded into this whole population of cells that all are responding to that specific antigen. And so in the case of SARS-CoV-2, the virus that causes COVID-19, these lymphocytes would recognize a specific protein from that virus and provide protection against that virus, again, through production of antibodies and cytotoxic T cells. Okay, so the other thing I guess I didn't mention on that slide in the title, I've got it that not only is the immune system responding to non-self, it also wants to distinguish between threats and antigens that aren't a threat, such as protein in our food or pollutants or pollen, things that we breathe in. We want our immune system to ignore those foreign antigens. And so when a virus enters a host, so this is showing how this adaptive immunity gets activated, and is illustrated for a viral infection, but you could imagine this is a vaccine coming into the host instead of a virus. The virus or vaccine gets taken up by these cells called dendritic cells, and these are the cells that really are the ones that determine if it's a threat or not. And so it's important that dendritic cells respond to a vaccine, basically get fooled into thinking that this is a threat, that this is an actual virus infection. That dendritic cell will then become activated. And once it's activated, it'll carry the antigens from the virus or the vaccine to the nearest lymph node here, where it'll activate T cells. So dendritic cells will activate the T cells. And then if these are helper T cells, they'll help activate the B cells that also recognize this viral antigen. B cells will go on to make antibody, and T cells, if these are cytotoxic T cells, they'll go out into the body and try and find infected cells in order to shut down the infection before it gets out of control. Okay, and I talked a little bit about threats. So how dendritic cells recognize pathogens as threats through different molecular structures that are associated with pathogens. And so vaccines, again, have to either mimic a viral infection in this case, so that the dendritic cell sees it as a threat, or if you're just using like a protein vaccine, as individual protein, you often include these additional components called adjuvants. And adjuvants are basically different molecules that are included in a vaccine that help stimulate the dendritic cell in the absence of pathogen replication, which it normally would be activated by. And so adjuvants are included in a vaccine, as I said, to activate the dendritic cell. They come in different formulations for decades. Aluminum salts have been used as a way to stimulate the dendritic cells. There's others that are kind of a oil and water emulsion. And then these other three are kind of like pathogen mimics. So they're molecules that mimic parts of the pathogen that would be recognized by the dendritic cell, viewed as a threat, and mount an immune response. So that's kind of in a nutshell how the immune response works, and kind of what we need a vaccine to do to stimulate an immune response. So next I'm going to talk a little bit about the virus, just enough to understand the vaccine targets against this virus. And so with the virus, again, it's called SARS-CoV-2. That causes the disease, and the disease is called COVID-19. It's a little confusing because there's two names. And the vaccines that I'll talk about are largely targeting the antigen, the SARS-CoV-2 antigen present within the spike protein. And so this is a SARS-CoV-2 virion, and these proteins that extend out from the surface of the virus are called spike because they look like little spikes. And they're embedded in a lipid bilayer. And so this virus has this lipid bilayer that surrounds and protects the viral genome, which is this kind of bead on a string structure within the particle. And so the virus basically just has to deliver its genetic content into a host cell in order to start replicating and infect that cell. And the reason spike is important, and we want to block spike, is because spike is really the first step in allowing the virus to enter a cell and infect it. And so that's shown here. You've got spike interacting with proteins on the host cell surface. So this is called its receptor protein, where that allows the virus to attach. Then different enzymes on the host cell help activate that spike and allow it to mediate this conformational change here, which is kind of like a harpoon, where the spike extends out its structure and stabs part of its protein structure within the lipid bilayer of a host cell. And so this kind of harpoon-like action brings this virus close to the cell and mediates a fusion event between the lipid bilayer of the virus and the lipid bilayer of the host cell. And that fusion allows the viral genome here to be released into the cell to start an infection. And so antibodies against spike are shown down here. They can bind to all different regions of the spike protein and inhibit this entry step. So whether it's binding to the part of the spike that interacts with the receptor to block attachment, or binding to parts of spike that prevent this conformational change, such as this one here, they're all preventing this whole entry event that needs to happen for the virus to start replication. And then on the right is showing where immune components against spike are important after a cell has been infected as well. And so if you have an infected cell here, once it's infected, it'll make spike proteins and put them out on the surface of the infected cell. And so, at that point, antibodies can bind to the spike and basically tag this cell. The antibodies will let other cells of the immune system know that this is infected, and those cells will kill the infected cell. And then this is how T cells recognize this is an infected cell. Basically, the spike protein, I don't have this drawn on here, but inside the cell, the spike protein will get chopped up into smaller antigen pieces. Those get displayed out here by host cell proteins that are recognized by the cytotoxic T cells, and they'll mediate killing and removal of this infected cell, again, before it can release any virus and spread the infection. And so all the vaccines we'll talk about are directed against the spike protein. If you've been following the news, there's been recent kind of news stories about a mutation in the spike protein that has spread and kind of replaced the original spike protein in the original virus. And so basically all proteins are made up of individual subunits, and so this mutation changes just one of those subunits in the spike, and this is where it's shown in the trimer. The spike is a trimer, so there's 3 of these subunits that get changed, and it's just subunit number 614. And the original virus that was originally identified in China had a D subunit at the 614 position. And then as the virus spread, there were new viruses, or there was a mutation in the spike that had a G subunit at this 614 position. And so you can see this kind of global transition of the amount of virus in the original, the original strain, the D614 virus. Sometime around February, it started getting rapidly replaced by the G614 mutation, viral mutation, and you can see this happened in the United States in March as well. And so this mutation, it again changes just one of these amino acid subunits in the spike protein. It's been found worldwide, and any place that it did arise, even in the presence of what we call the wild type or the original virus, that mutation eventually became the dominant strain in these locations. And so this happened really quickly, and so people really wanted to know what this mutation does. And so the newest paper out compared patients that had the G614 virus compared to the D614 virus. And with this mutation, these patients have an increase in viral load, so they have more virus when they detect virus in their respiratory tract, but they have, this has no relationship with disease severity. And so even though they have more virus, it's really not causing any worse disease when it has this mutation. And then important for vaccine development, when they looked at, they compared the G614 virus with the original D614 virus, and they showed that they're neutralized by antibodies equally well. And so any vaccines that were, all the vaccines in fact that were designed against this original virus should still provide protection against the new strain that has taken over. Okay, so now I'm going to talk about vaccine development. This is just kind of an overview slide. For the sake of time, I think I'm going to move on rather quickly. I'm going to talk about these different types of vaccines individually, so I'm not going to talk about them there. I guess I'm just going to mention over here is kind of the normal process of vaccine development, where you develop the product. It goes through various phases of clinical trials, so testing in humans. I will talk a little bit about or mention some trials. And so phase 1 and phase 2 trials are largely safety trials, just to make sure the vaccine is safe, it doesn't cause adverse reactions. And then also phase 2, they start looking at immune responses. So does it induce antibody and T cell responses? And then phase 3 is the final phase before FDA, gives licenses of vaccine. And these phase 3 trials are really large trials, minimum of like 30,000 people. And they're not only looking at safety and what immune components are activated, but also looking at whether the virus is effective or not. And so whether it prevents people from getting sick with COVID-19. And then usually they get approved, they start mass producing that as vaccines so they can be given to human. And one thing that's going on right now is kind of a collapse of phase 1 and phase 2 trials together, is basically jumping straight into phase 2 trials. And then having ongoing large-scale production of these vaccines that are still in trials, just in case they do show effectiveness and get licensed. That way we've got these billions of doses ready to go if they get the go-ahead. So these are all kind of compressed because of the pandemic and the urgency to get a vaccine. All right, so the next section, I'm just going to go through these different types of vaccines, how they're made, a little bit how they're made, and how they might induce an immune response. And so the first two are probably in development, not anywhere near clinical trials, and they're really not practical to the current pandemic. And so these are what I would call traditional vaccine strategies. Attenuated vaccines and inactivated vaccines. So a lot of the vaccines we get for other diseases fall into one of these two categories. An attenuated vaccine is just a weakened form of a virus, and so it can still replicate in the person that receives this vaccine, but it does so very lowly and doesn't cause disease. And so because it replicates, it induces a nice immune response. You don't need to trick the immune system into believing there's a virus there. The reason I say it's not really practical at this point is because attenuated viruses are made by growing them in different host cells, so non-native host cells, or at a different temperature over many, many passages. And this allows them, or causes the virus to adapt to these new growth conditions, and then they no longer replicate well in humans. And that's not really practical. As you can imagine, this virus needs to be worked with and grown in biosafety level 3 labs, which are the work is much more burdensome and expensive to do at this level. And then attenuated viruses are also made by genetic modification. But in order to do that, we really need to know the virulence mechanisms that would need to be eliminated to attenuate the virus. And that research just hasn't been, isn't far enough along yet. With attenuated vaccines, you also always kind of face a risk of reversion. So the attenuated virus reacquiring virulence in vaccinated hosts. And then there's a risk to immunocompromised patients. So they, the virus might be attenuated, but only in people with a healthy immune system. The next traditional vaccine is an inactivated vaccine. And so here you grow up large batches of the virus, treat it with either chemicals or radiation to inactivate it, but kind of freeze it in its native structure and use that as a vaccine. And again, with the inactivated vaccine, kind of the same problem. You have to grow up very large batches because you have to give a lot of it. It's not replicating, and so you need to give a large amount as a vaccine. So you'd have to grow up large batches, again, in the BSL-3 conditions and in cell culture, again, which is laborious and expensive. So the vaccine strategies, the rest of them are being used currently and tested against SARS-CoV-2. So the next one relies on just delivering a purified protein to a host, and again, in this case, again, it's the spike protein. This is the molecular structure, what the spike protein looks like, and of course the important receptor binding domain where antibodies would block its interaction with its receptor. So just producing the spike glycoprotein from a recombinant DNA type platform is much more rapid and easy for manufacturing and much safer as far as like compared to an attenuated vaccine. And then the other strategy here is because it's hard to get the immune system to see just a purified protein as a threat, you can make what's called a virus-like particle or VLP system. And this basically takes that spike protein and embeds it in a lipid bilayer so it looks more like a natural virus that the immune system would want to respond to. And so both of these, again, need a little help with immune stimulation, so including adjuvants, again, especially with the purified protein vaccines is absolutely necessary. And because they're not getting amplified within the person receiving the vaccine, these often require multiple doses, like up to like 4 booster shots even of these vaccines. And so that's kind of a drawback as well. But there are purified and VLP vaccines currently in phase 2 trials against SARS-CoV-2 by these companies listed here. And so again, they're still kind of in the safety trials. The next stage, or the next type of vaccines, are what we call nucleic acid vaccines. And so nucleic acids are DNA or RNA, and they would be given to a host to make their own antigen. And so they're going to encode the spike protein as the antigen. And so the picture over here, I'll just walk through to show you how this works, is you would have a DNA construct that encodes the spike protein, encase it in a little lipid bilayer or monolayer. The lipids allow host cells to take it up just like they would a virus. And then once the DNA is in that dendritic cell, in this case, the DNA would be used to transcribe messenger RNA or mRNA, which serves as the template for making proteins. And so the mRNA would be used to make the protein antigen, again, in this case, the spike protein, to activate immune responses. And then with mRNA vaccines, it's very similar, except you're skipping the DNA step and just delivering the mRNA straight to the cell, again, through a liposome here. and then the mRNA is used to make the spike antigen. And so these are again quite safe. They don't have any components from the actual virus except this one gene. Allows for rapid manufacturing, and so far in the trials that have been published so far, they induce both T cell and antibody activation or responses. I put effectiveness and longevity and with a question mark here because there are currently no licensed either mRNA or DNA vaccines for use in humans. So this is a really fairly new technology that we don't have kind of existing vaccines to tell us how well they might actually work. But there are a number of modifications that can be made to these vaccines to address that issue. And so time will tell how well these do. And so the top, the top vaccines currently that are in phase 3 trials already is an mRNA vaccine from a company called Moderna. And then Pfizer is also just getting started a phase 3 trial with an mRNA vaccine as well. And so again, we'll see soon if these are effective at protecting against disease. The next category of vaccines are basically taking the same concept as far as just delivering the gene that encodes the viral antigen, that encodes the spike protein, and introducing it into the host within a virus particle. And so again, this is going to be something that is better at tricking the immune system to respond to thinking this is a threat. And so there are two ways this is done. The one on the left is basically taking an attenuated virus or a replicating virus that doesn't cause disease. And so in the example, the attenuated virus is called VSV, vesicular stomatitis virus. And they've taken this attenuated VSV and replaced its surface protein here with the spike protein gene of SARS-CoV-2. And so they put the gene into the genome of this virus, and now the virus has the SARS spike on its surface instead of the VSV spike. And so again, it's an attenuated replicating virus, but it's going to have the SARS spike on its surface and no other genes from the SARS-CoV-2 virus. And so advantages of this, just like an attenuated vaccine, it's going to amplify within the host. So it's going to be given as a single dose. And the ones in trial are given orally. And so you don't have to worry about needles or anything. So currently Merck has a phase 2 trial right now with one of these. It's in a measles vector backbone for the attenuated measles that we get vaccinated against. They also have one in preclinical trials with the same VSV backbone. And then the other strategy that's being used as a kind of a viral vector strategy is to use a non-replicating virus. And so this is really just in packaging. And so they take the gene that encodes the spike protein, and this is the only gene in the virus. And they put that gene inside this kind of virus shell. In this case, it's an adenovirus shell, so it looks different. It doesn't have a lipid bilayer. It's just this protein shell. And that delivers it to the dendritic cells, which will then express the spike protein and deliver to the immune system. They're using adenoviruses here. One is using a chimpanzee adenovirus. One is using kind of a rare adenovirus strain. And this is important because if they need to choose viruses that we don't already have antibodies against, because if we have antibodies against this vaccine structure, it won't react. It'll get eliminated by our existing immune response. And so there's two of these again already in phase 3 trials You may have heard of the AstraZeneca one because it's getting some bad press. This is one, the phase 3 trial is on hold currently in the United States. There's been a couple of adverse reactions. So 2 people that are in the trial have inflammation in their spinal cord. The first one, they ended up, the person was diagnosed with multiple sclerosis, and they said it was unrelated to the vaccine. But then when the second one happened, they paused the study. It's been restarted in the UK and Brazil, but so far the United States still has it on hold while they're investigating whether these are adverse events due to the vaccine or not. Okay, so I wanted to talk a little bit about other things that have hurried vaccine development at kind of this unprecedented rate for SARS-CoV-2. So some of this, well, a lot of it, and the reason why we have several of these vaccines that are already in clinical trials is due to basically taking vaccines against other viruses that are currently being studied or have been licensed, and just plugging in that spike antigen from the SARS-CoV-2 virus in order to get it through clinical trials faster. Like if they've already been through clinical trials in one form, just replacing that one antigen allows those trials to go through more quickly. And so some of this is based on ongoing vaccine development against other pathogenic coronaviruses. We talk a lot about SARS-2 or COVID-19, but it's SARS-2 because there was an original SARS coronavirus that emerged in humans in 2002 and spread worldwide, definitely not to the levels of COVID-19. But it was, the pandemic in that case was stopped due to public health measures. And so the vaccine production pretty much halted at that point because the virus no longer was a threat. And then another pathogenic human coronavirus, the MERS coronavirus, emerged in 2012, and it's still present in human populations, and we're still seeing infections with this virus in the Middle East, but it's not as effective at spreading human to human as the SARS or the SARS-2 viruses. And so vaccines that have been being developed against MERS especially also has spike, some of the same mechanisms, and so just swapping out the new spike for that MERS spike has allowed these vaccines to be developed more rapidly. They've also built upon kind of vaccine technology that's been developed for other unrelated viruses. So I mentioned the VSV vectored spike vaccine that Merck is going to be testing in human trials soon. It's based on their licensed Ebola virus vaccine. So they've got a vaccine that has that VSV with the Ebola virus spike protein on the surface. Again, other viruses that are currently kind of a threat or have been recently, so their current vaccine is being tested against them, but not licensed yet. Those platforms have been kind of harnessed for making SARS-2 vaccines, or dengue virus is another one that's a very new vaccine we've got that decades of research challenges went into generating that vaccine, and so learning from those studies has been important. And then with these newly developed vaccine platforms, DNA vaccines, again, we don't have any licensed in humans, but there's a West Nile virus vaccine for horses, and then the mRNA vaccines that have been in development for a while allow a really rapid vaccine production as far as, again, just using the same platform and just changing the gene of interest. Okay, I want to talk a little bit about a couple kind of outside the box thinking strategies, because I think they're definitely interesting compared to traditional vaccine strategies. The first one is from a company called Ivida Biomedical, and they're currently in a phase 2 clinical trial with this vaccine. And what it does, the vaccine, they basically take dendritic cells out of an individual, grow them in the lab, and activate them and load them with SARS-CoV-2 antigens, and then take these activated dendritic cells and inject them back into the individual to mount an adaptive immune response against the virus. So this one, I had to read it a couple times because I was kind of stunned that was a strategy in phase 2 trials, because it seems like it would be really, really slow process doing this for everybody that needs a COVID-19 vaccine. But what's interesting is it's really this company, this is kind of their first foray into infectious diseases that I can see. This is a strategy they're really using for kind of a personalized medicine approach to cancer immunotherapy. So their phase 2 vaccines that they're are known for have this approach, but they're also taking tumor cells from this individual. And so then the antigens they're giving are special, are specific for that individual's tumor. And so it's an awesome strategy for cancer immunotherapy, and kind of interesting that they're applying it to infection. And then the next out-of-the-box strategy is using attenuated vaccines for other pathogens, so completely unrelated pathogens, as more of an immune stimulant. So these have no SARS-CoV-2 antigens in them. So they wouldn't induce any kind of B cell and T cell adaptive immunity against SARS-CoV-2. But the hopes is that they would stimulate kind of the innate immune system to either help eliminate the virus or control the virus early in infection, or lead to a faster adaptive immune response in someone who gets infected. And so the two that are being tested, they're currently in phase 3 trials because they're already approved vaccines. One is against poliovirus, it's the oral polio vaccine, and one is against tuberculosis, and so it's the BCG vaccine. This one given orally, this one given by a shot, but they're both attenuated viral vaccines. And you know, the question is will it stimulate immunity in the respiratory tract, as neither of these pathogens are, or neither of these vaccines, tuberculosis is respiratory, but neither of the vaccines are given through the respiratory route. And so I'm anxiously awaiting the results of these trials because I think it's kind of fascinating. Okay, so the last part of my talk, I want to leave plenty of time for questions. Is, okay, say one of these vaccines works, we get approval, and we get the vaccine into human populations. How many people need to get vaccinated for it to provide herd immunity against COVID and make an impact on the actual pandemic? So a couple weeks ago, we had Bert Baumgartner and his gang talked in the colloquium about vaccine uptake and people's views about whether they're, or I guess, willingness to get a vaccine or not. And some interesting numbers have come out in these this Pew Research Center that does surveys. They surveyed US adult populations and asked them if they would be likely to accept a COVID-19 vaccine if one was available. And in May, when they sent this survey out, 72% said they would be willing to get a vaccine. And in September, this has dropped to 51%. So we're definitely seeing reluctance. You know, this could be a whole nother Renfrew colloquium on the kind of rushing of vaccine trials and and the politics that are driving vaccine approvals, and it's really having an effect on public trust. So I want to talk just a little bit about herd immunity. We've heard it a lot, H-E-A-R-D, it a lot, but how does it actually work? And so these different pop squares are supposed to represent different populations of people. The blue circles are people who are unvaccinated or have no immunity against the virus. The yellow have immunity in this example because they've been vaccinated. And then the red dot in the middle shows kind of the introduction of a single infected person within each of these populations, and how their contacts with susceptible people changes based on the level of immunity in a population. So you can see as you get up to like 75% of the population is vaccinated or yellow, there's fewer contacts between this person with the blue or the unvaccinated people. So if there's a video, hopefully it works, it'll kind of show how virus might spread from this person in these different populations. All right, so I'm glad that worked. But you can see a real dramatic difference as you increase the vaccination or immunity rate and how the virus is able to spread through the population. And this is based on, the mathematicians and modelers can estimate how much of the population needs to be protected in order to provide herd immunity. I'm not going to go in through the details of transmission and that's shown on the slide for sake of time. But basically, if you estimate this is an index of how transmissible a virus is or pathogen, and then how much of that population would need to be vaccinated to have herd immunity. And COVID is estimated to fall somewhere in between Ebola and polio here. And so you can imagine that somewhere between 50 and 80% of the population would need a protective vaccine in order to really stop the pandemic through vaccination. All right, so I'm going to wrap up here with just some kind of upcoming challenges and hopefully a silver lining. So the first challenge I haven't mentioned yet is sterilizing immunity. So this is really going to affect that herd, the level, whether herd immunity by vaccination is effective or not. And so sterilizing immunity just means that a vaccine would prevent a person from becoming infected so they couldn't transmit the virus. Really the kind of goal of the vaccines that are being tested is not sterilizing immunity. The goal is just reducing disease severity. So we might, it's very likely we'll end up with a vaccine that reduces the severity of the disease, reduces your chance of dying from COVID, but doesn't stop transmission. And so in that case, there's no herd immunity even on the table. I've mentioned long-lived protection. That's a big question really with all of these vaccines, and we can't tell that from these short clinical trials that are taking place currently. The vaccine is going to need to protect people that are most at risk for severe disease, so older adults and people with comorbidities or immunosuppression. And so it needs to obviously be effective in these populations. The vaccines, some of the vaccines that they're testing have to be kept at ultra cold temperatures, so colder than your average freezer. And so getting them shipped and to pharmacies and to clinics and to patients is impossible in parts of the world that don't have this good supply cold chain that we have, but it's even going to be difficult in the US if they have to be at ultra cold storage conditions. Talked about public trust, that's going to be a huge obstacle. And I think we also, kind of a good and bad, that we're seeing from this surge of vaccine development is that if we have support for vaccine development between pandemics, some of these platforms would already have been up and running and would move even faster when a new pathogen is recognized in the human population. And so an example of this, the mRNA vaccine platform, they've generated these mRNA vaccine, Moderna, from the viral RNA sequence, from knowing the sequence of that virus to human trials took 63 days. When most, or the average, I guess, of getting a vaccine after a pathogen is discovered averages 10 to 15 years. So there is this, tremendous speed at which these have come about because these platforms were already being studied. All right.